Hello everyone, I'm Abhinav Banerjee, a PhD student from Dr. Mahipal Ganji's lab at the Department of Biochemistry in Indian Institute of Science. Our lab mainly focuses on developing new single molecule techniques to study biological phenomena and to also develop novel DNA paint probes to extend the ability of DNA paint super resolution microscopy. We would like to thank TNQ Technologies for honoring us with the Inspiring Science Award for the best paper published in India in the field of biological science for the year 2023. I am here to briefly explain our work and to talk about how our research findings have shed new light in the field of biological sciences. We have measured base stacking interactions using DNA nanostructures. We know that DNA is a double helical molecule where two different DNA strands are present in a helical manner intertwined between each other. This molecule is stabilized by two different types of interactions. Base pairing interactions between these two DNA strands occur between A and T or G and C and are very crucial in bringing these strands together. A rather less talked about interaction, which is very crucial for maintaining the structural stability of this molecule, is the base stacking interactions that happen or that occur between two adjacent bases on the same DNA strand. Though these interactions are extremely weak in nature, they are very essential in maintaining DNA's double helical structure. To measure these weak interactions, we break down a double-stranded DNA into two different configurations. The gap configuration does not have base stacking interactions between this red and the magenta strand, and a nick configuration where there are base stacking interactions between the magenta and the red strand. To bring DNA strands in such a configuration or in such a structure, we exploit the use of DNA origami. DNA origami is made using single-stranded DNA in a predictable manner, where a long single-stranded DNA, which is shown in gray color here, can be folded into a shape that we desire using these differently colored short single-stranded staples. As the position of these staples are designed by us, we can very precisely position them in such a manner to bring two adjacent staples carrying defined DNA sequences at close proximity to each other. We also use DNA paint super resolution microscopy, which relies on DNA complementarity again. Here, a single-stranded DNA attached to the target is visited by its complementary strand that carries a fluorophore in a very periodic manner due to its complementary nature. When these strands visit the target strand, we get signals as in an on and off pattern, which can be clearly seen in this video. Each of these signals can then be fit using different localizing algorithms to achieve super resolution imaging. Here we see DNA origami structures where each of these spots are separated by 20 nanometers on a DNA origami that is 90 by 60 nanometers in length and breadth. Coming back to our DNA origami assay, here at the assay site, we have these two different configurations, one without the stacking interactions and one with the stacking interactions. We observe that the imager binds at for different durations of times. In the absence of stacking interactions, it binds for very short durations of time. But in the presence of stacking interaction, the duration the imager binds increases dramatically. Based on what different dinucleotide pairs are present in the vicinity at the stacking interaction zone, we can see that if there are AC nucleotides present, the enhancement is almost 250 folds as compared to CT, which shows only 6-fold enhancement in the binding time. This shows that different dinucleotides can show a wide range of base stacking interactions, and using very simple thermodynamic calculations, we can extract the free energy that each of these dinucleotide interactions show. We further extend our studies to assemble higher hierarchical multimeric DNA origami structures, where we use a 3-point star to assemble a tetrahedral DNA origami structure. Here at the interface, if we can, uh, we change the base stacking interactions as shown here, where we can change it either as AC, GA, or CT. And we see that when AC interactions are present, the structure folds to be the strongest. And when CT interactions, which were the weakest base stacking interactions, are present, the multimeric DNA origami structure never folds and are undetectable in our study. We are using this information of the stability gained or the stability imparted by the base stacking interactions to the imager for binding longer to develop new DNA paint probes that can actually detect two protein molecules which are present at close proximity to each other. We are naming this proximity paint, and we are also developing new DNA paint strategies to develop multiplexed imaging where we can image different protein molecules in the same sample, where the base stacking interaction contributed by these different targets would vary, and thus we can decipher which target we can image multiple different targets in the same cellular sample. I hope this brief explanation was clear to everyone. Please feel free to reach out on my email, which is available on our lab website. Thank you, everyone, and have a great day.